Welcome back to week 8 of Introduction to Complex Biological Systems. So today I am going to discuss protein evolution. So this week I am discussing evolution, the history of life. So today I am going to discuss protein evolution. How do we study evolution? Right. So let's see at one example. We normally study evolution based on fossil records that we get. Now here you see three different bones are shown. Okay, so these are fossils. So this is from a female chimpanzee, this is from a human female, and this is from some other species. Okay, so if you look at this particular bone, you will see that the length of this bone is something similar to this. However, when you place this bone on a horizontal surface like this, it tilts towards the right, just like the human bone. Okay. This is something that is typical to us, but if you keep a chimpanzee bone, this is the femur, it will stand vertically like this. Now, this tells you that whatever this species is, it sort of shares a characteristic between this and this, right. So, these two species, and we know when chimpanzee appeared and when the first human species appeared, right. So, this is something that is in between, and then if we can do carbon dating to know when this particular, how old this fossil is, right. So, this is actually one of the most famous fossils in the world. It is 3.2 million years old partial skeleton called Lucy. Okay. So, partial skeleton means that almost 40% of the skeleton, the complete skeleton, was recovered in this particular dig. So, the fossil Lucy refers to this partial skeleton of an early hominin species. Okay. So, it is called Australopithecus afarensis. So, A. afarensis. Okay. It was discovered in 1974 in Ethiopia by Donald Johnson and his team. So, this is how we sort of study evolution, that we look at this fossil record. Based on the height of this, we can say that this species, they were not as tall as modern humans, but they were somewhere in the same range as these chimpanzees, right. However, this tilt tells you that they can walk upright like us instead of the chimpanzees. So, chimpanzees, they cannot walk upright like us, but this hominin species, she can actually walk upright. Okay. But however, from the fossil record, from the skull, you can tell that the brain of this species was not very big. So, it was not as large as the modern human. It was somewhere similar to this modern chimpanzee. So, you can also conclude that the evolution of walking upright evolved earlier than the evolution of increase in brain size, right. So, this is how we can draw all these different conclusions. Now, protein evolution can be studied in a very similar manner. So, we can compare two proteins which have the same structure. So, we are comparing the same bone here, right. We are not comparing different bones, we are comparing the same bones here. So, similarly, you have to compare the same proteins from different species, and then by looking at how different they are, we can sort of tell whether which one appeared first. Okay. Or if we have two different proteins and we find something that is in between, we can say that if this is modern, this is older, this will be somewhere in between, because it shares trait with both the proteins, right. So, that is something that we do for proteins. So, let us see how we do that. So, I will take up the example of hemoglobin and myoglobin. So, I have talked a lot about hemoglobin. Hemoglobin is the protein that carries oxygen. So, it has this quaternary structure, 4 different polypeptide chains are there with the heme group. So, there are 2 alpha chains and 2 beta chains. The monomeric form of this protein is called myoglobin. So, it has only 1 polypeptide chain, it has 1 heme group, it also binds oxygen. If we compare the amino acid sequence of hemoglobin and myoglobin, so these are the 2 chains of hemoglobin, the alpha chain and the beta chain, and myoglobin. So, we have written all the amino acid sequences here, right. it turns out that only 28 out of 150 residues are identical in these proteins. So, identical means that this position all are methionine, all are leucine, all are valine, like so. So, if you count, there are only 28 amino acids like this which are same in all these 3 sequences out of almost 150 amino acids, right. So, only 70% identical. It means that all the other positions can change, but if you look at the structure, they look very similar. So, there are exactly the same number of helices. So, I think there are 7 helices, right. So, there are same number of helices in them, and if you superimpose the structures, so take one of this subunit and superimpose with this, you will see they superimpose quite nicely. So, even with only 70% identical residues, the proteins fold in a similar manner. It means that we have to look for something beyond the identical residues. Okay. 45% of the residues are identical between the alpha and beta subunits of hemoglobin. So, if we ignore myoglobin, if we take only these first two, you will get this V, you will get this P, right, you will get this K. So, you will get some additional residues. So, these two are K, but here it is W. So, you will get these additional residues, and it turns out that it increases from 17% to 45%, but still it is less than 50%, right. Sequence identity between alpha and beta subunits of hemoglobin and myoglobin is only 30% and 26% respectively, which means that if I remove beta, if I just look at alpha and myoglobin, that will be 30%. If I remove alpha, if I look at beta and myoglobin, that will be 26%. Okay. So, the best that we see is 45% sequence identity. So, it means that we also have to look at something else apart from identical residues, and that is called similarity. So, what I mean by similarity? So, for example, if I look here, hemoglobin alpha chain has a valine, beta chain has a leucine, myoglobin also has a leucine. Now, if you look at the chemical structures of valine and leucine, they are very similar, only one carbon difference, right. So, both are hydrophobic, both have 2 methyl groups. So, you can imagine that we can easily replace, or we can more often replace a valine with a leucine or a leucine with a valine. So, valine and leucine will be residues which are similar. Okay. Similarly, if I think about this, so here in hemoglobin it is glutamic acid E, and in myoglobin it is aspartic acid D. So, both are acidic groups. So, if you replace one with another, you are not going to change the characteristic of the side chain. So, that will be also, these will be also similar residues. So, you can replace one with the other, right. Similarly, if we replace phenylalanine with tyrosine, then they will be similar. Okay. So, we can group residues like that according to the chemistry of the side chain as similar residues. So, now let us look at myoglobin, hemoglobin alpha chain and beta chain, again similar protein from other organisms, right. So, there are total 8 different protein chains from these different organisms. If you look at the structure, they look exactly the same, right. Now, we can align their amino acid sequences and see how similar or how different they are. So, if I do an alignment of all these 8 amino acid sequences, this is something that you will see. So, only 2 residues that are shown here are absolutely conserved. So, this is a histidine. So, it is histidine in all the sequences, and this is phenylalanine in all the sequences. Everything else can change, and they are color coded. So, anything that is in darker green means it is more conserved. So, you see that it is mostly proline except Q here. This is mostly tryptophan except in 2 positions it becomes phenylalanine. Here it is either valine or isoleucine. So, almost 50-50, right. As you go lighter and lighter, so in this case valine, isoleucine, nothing, right. Valine, isoleucine, phenylalanine. So, hydrophobic residues, but still it changes from methyl group to aromatic group. And then there are positions which are white. So, it can become anything: leucine, phenylalanine, histidine, threonine, aspartic acid, isoleucine, asparagine, valine. So, all 8 are different, and that is why it is white. So, from this you can tell that there are many positions where you can completely change the amino acid without any consequence on the structure of the protein. In fact, there are regions where you can delete amino acids and still the protein folds in the same manner. So, this poses an interesting question that how do they do this sequence alignment? How do you know what aligns with what? So, here it is exactly identical. So, I know that this is identical, but then you are putting a gap here. So, instead of aligning this aspartic acid here, I can put this aspartic acid here, right. But I have not done that. I have put 3 gaps and then I have put the aspartic acid here. So, this histidine I can easily put here because this histidine comes after this serine. So, I have not done that. I have put this huge gap and I have put this histidine here. So, how do you do that? How do you know which residues are aligning with each other, where you have to put these gaps? And so, I can align 2 residues like this, or I can align them like this, or I can align them like this, right. So, how do you align these residues? Okay. And can we measure this? Can we quantify how similar 2 sequences are? So, this will be something that will be very useful for further studies, right. So, now before I do that, let us just point out why these 2 residues are conserved. So, if you look at the structure, this histidine is the one which directly coordinates with the iron, and histidine is the only residue which can fit there and which can do this coordination. So, you cannot change this particular histidine with any other amino acid. So, that is why this histidine is absolutely conserved. On the other hand, this phenylalanine, which is also conserved, shows up here, and this is also conserved because this phenylalanine allows the oxygen binding and binding in the correct orientation. If I change this phenylalanine with a tyrosine, which means I will put an oxygen group here, then there will be no place for this oxygen binding. If I change it with tryptophan, the structure will change, right. So, no other amino acid will be tolerated here because of this very strict requirement. That is why phenylalanine is absolutely conserved. So, histidine and phenylalanine, these 2 amino acids are absolutely conserved. The remaining amino acids you can change, but again that change cannot be random. Okay. So, there are certain characteristics that have to be preserved at certain positions. So, let us look at in more details how can we compare 2 sequences. So, what I have done is I have given 2 sequences. So, this top one is from protein 1, this bottom one is from protein 2. Similarly, again the top one is from protein 1. So, it is the exact same sequence shown up here. This bottom one is from protein 3. Now, the question that you can ask yourself is whether protein 2 is more similar to protein 1 or protein 3 is more similar to protein 1. So, which one is more similar to protein 1, protein 2 or protein 3? So, let us say these 3 sequences come from the same protein, but from different species. So, based on that we can also answer whether species 2 is more similar to species 1 or species 3 is more similar to species 1, right. So, if you look at this, here only one amino acid is identical, everything else is different, and in this case 3 amino acids are identical, everything else is different. So, if I go by sequence identity, then only one is identical here and 3 are identical here. So, based on identity, I will say protein 3 is more similar to protein 1 than protein 2. Okay. So, protein 3 is more similar to protein 1. But will that be correct? Because we also have to take into consideration something called similarity. So, if I look at this, let us say asparagine and serine, both are polar residues, but here asparagine is changed to an arginine, which is a charged residue. Arginine to lysine, both are positively charged. Arginine to leucine, so positively charged becomes a hydrophobic residue. Okay. Leucine to isoleucine, leucine to proline. Proline is it's a amino acid, right? Very different structure. So, alanine to methionine, both are hydrophobic. Alanine to aspartic acid, which is negatively charged. So, we have to take into consideration these other substitutions also. So, if we do that, there are these numbers which are obtained for a pair of substitution. So, 0.71 for substituting alanine with methionine, 1.41 for substituting valine with leucine, so on and so forth. Now, these same numbers are converted to, if you take the log, so log of this, so 2 log to the base 2 of this L gives you this S, right. Now, if it is this, you have to multiply all of them and you get something, but instead of multiplying numbers, if I take the log, then I can just add them, right. Addition is computationally easier compared to multiplication. So, that is why people convert this into a log scale, so that you can just add these coefficients and get some number. So, protein 1 and protein 2, if you do this, you get this number as 13. What do we get for protein 1 and 3? So, if I do that for protein 1 and 3, I get this number as 2. So, clearly this is greater than this. So, higher this number, more similar these two proteins are. So, it means protein 1 is more similar to protein 2 than protein 3, right. So, even though we have more identical residues here, but if you take into consideration similarity, then protein 1 is more similar to protein 2 than protein 3. So, now the question comes that how do we get these numbers? How can we tell that, okay, what is the weightage of substituting one amino acid with another amino acid? And this type of weightage is given by a matrix called the Blossom matrix, block summation matrix, right. So, if you look at this matrix, it gives you what is the weightage of substituting one amino acid with another amino acid. So, if I take serine, if I substitute a serine with a cysteine, it gives me -1. So, 0 means there is no preference for this change. So, it can happen. So, there is a random chance of mutation of a serine to cysteine, right. If you have this number as 0, it means that this substitution is observed which equals to this random chance. If the number is negative, it means that this substitution happens less often than what you would expect by random chance, okay, which means that this substitution is unfavorable. Okay. Since it is unfavorable, we see it less often. If it is positive, then this substitution is favorable. Favorable in the sense that these two residues are very similar. So, the change between them happens more often than you would expect by random chance. Okay. So, let us look at some numbers. Glycine versus cysteine. You see, glycine versus cysteine is -3. So, cysteine has this side chain SH, glycine is the smallest amino acid. So, glycine will not be changed by a cysteine, or a cysteine will not get changed by a glycine. That will be detrimental to the protein. So, glycine cysteine or cysteine glycine mutation is seen less often than you would expect by random chance. On the other hand, if you see this is 3. So, valine to isoleucine. Both are hydrophobic side chains, both have 2 methyl groups. So, both are hydrophobic amino acids, both have methyl groups as their side chains. So, substitution of an isoleucine with valine or substitution of a valine with an isoleucine is seen more often than you would expect by random chance. So, that is this positive number that signifies. Okay. So, I have marked some of them here. So, for example, isoleucine with leucine and leucine with glutamic acid, aspartic acid. So, this is negative, this is positive. So, you can explain yourself why this is negative and why this is positive. You will also see a number in the diagonal. Now, this 11 means changing tryptophan with a tryptophan. Okay. So, of course, you are not mutating tryptophan with a tryptophan. So, why we have this number in the diagonal? The number in the diagonal signifies how hard it is to mutate tryptophan into something else. So, higher this number, it means more difficult it is to mutate this particular amino acid. So, tryptophan is the one which is the hardest. Then it is cysteine because it can form disulfide linkage which no other amino acid can, then it is histidine, which has a very unique pKa for its side chain. Okay. So, frequency of amino acid substitution is something that is given by this type of block substitution matrix. Now, so how do you determine these numbers? To do that, what is done is a set of alignment of sequences of proteins that are related to each other, right? Like we saw the example of hemoglobin, myoglobin and all these other proteins. So, globins, or one can take triose phosphate isomerase enzymes or some other enzyme from different species. So, you get this bunch of amino acid sequences, you align them, right? You break these alignments into smaller blocks that are uninterrupted by insertions or deletions. So, there are no gaps. So, you take such blocks and there you measure the frequencies of this amino acid substitution, which I will show in the next slide. Now, one of the factors that is done is a threshold level of 62% sequence identity is used, which means that no two sequences will have more than 62% identity, okay? Because if there are sequences which have more sequence identity, then they will bias your numbers, your statistics. So, sequences are taken which do not have more than 62% identity, and that is why this is called block summation 62. So, this number was arrived by trial and error, and this was found to be something that works very well. Okay. So, let us look at this alignment again. So, we have taken this 8 sequences and we have aligned them somehow. Okay. Now, we want to look at places where there are no interruptions. So, I can look at this. So, I can take this block, so up to this and this. So, I can take this block where there are no insertions or deletions. I cannot take this region because there are so many gaps here, right. Similarly, I can take this region. So, I can go from here to this phenylalanine up to this. I can take this block, there are no insertions or deletions. So, let us say I take that. Okay. So, it will look like this. Now, we have to look, we want to determine the statistics for a pairwise substitution. So, let us say I want to look at the rate of substitution of phenylalanine with leucine or vice versa, leucine with phenylalanine. So, I will look at all these places. So, there are no leucine or phenylalanine here, no leucine or phenylalanine here. So, in this place I do see phenylalanine and leucine. Here I do see leucine, but it is not substituted by phenylalanine. In this case I do see phenylalanine, but there are no leucines, right. So, here I have leucine, but no phenylalanine. So, I can take only those columns which have both leucine and phenylalanine. That is why I can get the pairwise substitution. For example, if I look at this, there are other residues, but I do have 2 phenylalanine and 1 leucine. In this case, everything is phenylalanine, no leucine, so I cannot take this. So, these are the 3 columns that I will take which have both phenylalanine and leucine, and from here I will calculate the frequency of substitution of these 2 amino acids and then compare it with random chance and then see whether it is more than that or less than that. So, substitution likelihood ratio is that L, Ij are the 2 amino acids. So, it will be phenylalanine and leucine. So, it is given as the ratio of the frequency of an Ij substitution. So, substituting L with F or F with L in the alignment block of related proteins. So, this is my alignment block. Frequency of this divided by frequency of this Ij substitution in the same block, but with the position of all the amino acids scrambled randomly. So, if I take all these amino acid sequences and then scramble them randomly and then I calculate the L and F substitution, so that will give me the denominator. So, these are the 2 numbers. How do I get this F, the frequency of substitution in this alignment block? So, I will do this by counting the number of times I type and J type occur in the same column. So, how many times phenylalanine and leucine occur in this column? Okay. and then the total number of amino acid pairs in the sequence block. This Pij I will calculate like this. So, I will calculate it as 2 times Pi times Pj. So, what is this Pi or Pj? This I will calculate as number of occurrences of the ith amino acid divided by the number of positions in the alignment. Okay. So, how many times the amino acid shows up divided by the total number of positions in the alignment. So, let us look at the substitution of phenylalanine with leucine or leucine with phenylalanine. So, let us calculate this first. PL is 18 divided by 8 times 24. So, there are 8 rows and 24 columns. So, that is the total number of amino acids there. Okay. How many times my leucine shows up? If I count here, you will see it shows up 18 number of times. And what about phenylalanine? It shows up 25 number of times. So, these numbers are given here. So, 18 divided by 8 times 24 and 25 divided by 8 times 24. So, the denominator is exactly the same and you get these two numbers, right. So, those two multiplied by 2, you get this PLF, which is the denominator. Now, we have to calculate the numerator, which is this F. Number of times the ith type and the jth type occur in the same column. So, if I look at this column, 1, 2, 3, 4, 5, 6, there are 6 phenylalanines and 2 leucines. So, if I calculate the number of pairs, 6 times 2, that will be 12. Similarly, here 6 times 2, that will be 12, and here 2 phenylalanine and 1 leucine. So, this is one pair and this is another pair. So, 12 plus 12 plus 2. That is what I get, 12 plus 2 plus 12, right. That is the numerator. Now, I have to scramble the sequence. So, what are the total number of scrambled? If I do that, so what are the total number of pairs that are present, right. So, the total number of pairs that are present will be, there are 8 rows and 24 columns, right. So, 8 times 8 rows. Now, in each column there are 8. So, if I take this, this will be one pair, this will be another pair, this will be another pair, like that. So, 8 times 7, that is 56 will be the number of pairs in each column, and 56 times 24 will be the total number of pairs in this whole block. Now, I can calculate this QE and EQ, but they are the exact same pair. So, I am counting them twice. So, I have to divide it by 2, and that is exactly what is done here. 8 times 7, that is 56. So, that is the number of pairs in each column, and you divide it by 2, and then you multiply by the total number of columns. So, that becomes 672. So, 26 by 672, that is 0.387. Okay. So, this is the frequency which is the numerator, and this is the frequency which is the denominator. So, if you take the ratio of these two, you get this LF, which is 1.59, which is greater than 1, which is greater than 1. And if we take the log to the base 2 of this, multiplied by 2, then we will get S. So, it turns out that when we align two sequences, if the sequence is significantly long, more than let us say 100 residues, and we end up with 10 or 20% similarity, chances of these two sequences to be structurally similar is very likely, right. Because you have two sequences which are 100 amino acid long and their sequence similarity is 20%, that happening by random chance is very less. Okay. So, it means that they most likely are going to have very similar structures. So, this is the basis of something called homology modeling that people do for newly identified sequences, that they try to predict the structure for this new sequence based on the structures of sequences of proteins whose structure are already known. Okay. So, I mean, we can do this type of calculation to show that when two sequences have 20% or more identity, then chances of them being different is very, very low. So, let us consider a protein with 150 amino acid, right. So, if you consider its gene, it will be 450 bases. Now, there are 4 types of bases. So, what are the all possible sequences which will have 450 bases? That will be 4 to the power 450, which is 10 to the power 270 possible sequences, right. That is a huge number. If we just consider the amino acids, 150 amino acids, then a protein, or how many different 150 amino acid sequences we can generate, that will be 20 to the power 150, which is 10 to the power 195 possible sequences. Now, compare this 10 to the power 195 number with the total number of atoms, which is estimated number of atoms in the universe, which is 10 to the power 79. So, you can imagine that this is a huge number. Now, if two random sequences are taken and they have 10% or more identity, then that identity arising from random chance is going to be extremely low, which means that these two sequences originated from a common ancestor. So, there was some common protein in the past and then it diverged, accumulated mutations, and then after several million years, we are looking at these two proteins and the sequence identity between those two proteins is 10% or 20%, then you know that these two proteins originated from the same common ancestor. So, this is something that we use for creating something called a phylogenetic tree and also measuring time in the past. So, evolutionary time. So, a phylogenetic tree will look something like this. This tree has been constructed using a particular protein called cytochrome C. So, we have already seen cytochrome C in photosynthesis and respiration, right. So, the tree, the way it looks like is that this is the ancestor. At different points of time, this branching happened, right. Now, anything that is close, they are very related. So, they diverged at this point. This and this diverged much more in the past, right. So, things which are more separated diverged more and more in the past, things which are close together diverged in the near past, right. And these numbers are normally, they are some weighted numbers. You can also put them in terms of millions of years. So, phylogenetic tree are created using proteins which have certain criteria. So, for example, cytochrome C, it fulfills most of the criteria that are needed for creating a phylogenetic tree. So, it has this orthodox pathway of gene during evolution. Okay. So, nothing fancy, nothing funny happened. Okay. So, we can trace this protein in different species. Singularity of a gene. So, there is only one gene for this protein, not multiple genes like ribosome. Same biological function in all compared organisms. It is not doing something different in different organisms, right. It is found in all these different taxons of life. Cytochrome C is small, it has only 110 amino acids, which means that the sequence alignment will not be computationally problematic, right. So, you can actually do that meaningfully. So, this type of phylogenetic tree help us to determine how long back in the past two species diverged, right. So, whenever we talk about this time, it means that we are talking about some clock. So, where is this clock coming from? This clock is coming from the rate of change or rate of evolution or rate of mutations that happen in certain genes. If we know the rate of mutation, if that rate of mutation is not changing much over time, then if you compare two sequences and see how many changes have happened, you can sort of get an estimate of time that okay, it took this much time to gather this many mutations. So, this is something that is referred to as a molecular clock. So, molecular clock is based on the rate of molecular evolution, which can be remarkably constant over time. So, this is one of the major hypotheses of a molecular clock, right. This constancy of rate was explained by the neutral theory. So, what is a neutral theory? It states that most changes to DNA or protein sequences are neutral. That is, they are driven by drift, not by selection. Okay. So, now there are of course problems with this, but it is something that helps us in determining some of these time periods. So, of course, this molecular clock is an imperfect clock, right, because the rate of molecular evolution is influenced by mutation rate, which can be influenced by so many different things, right. So, pattern of selection, population size, stochastic fluctuations in this substitution rate. So, just by chance there can be accumulation of more mutations in a small period of time, right. So, all these things can make this clock really imperfect and the estimation of the dates imprecise. Variation in rate between different lineages can cause substantial bias in molecular date estimates. For example, if you consider unicellular organisms, evolution or mutation happens faster in unicellular organisms compared to multicellular organisms like us, right. So, the rate of mutation will not be same, and this variation can create substantial bias in the molecular date estimates that we are doing using this molecular clock. The basic approach for estimating molecular dates is to take measure of the genetic distances between species, then use a calibration rate to convert the genetic distance to time. So, this is the basic premise, right. However, of course, as pointed here, there are several problems with this. So, there are many statistical methods by which people do this. So, there are sophisticated methods like maximum likelihood or Bayesian approaches which estimate molecular dates along with other parameters of model of the DNA substitution process. Now, the reliability of all these molecular clock methods depend on the accuracy with which genetic distance is estimated and on the appropriateness of the calibration rate. However, this is still a challenging problem, but it still gives us some really meaningful and interesting insight into the evolution of these different organisms and species over this period of several billion years. So, for this lecture you can follow these books as I have listed here. Thank you.